I want you to imagine your body right now, but not as a collection of static building blocks.
Right, because that's how we usually picture.
Exactly. I want you to imagine it as a bustling, microscopic postal service. I mean, every single second, all throughout your tissues and fluids, your cells are throwing out millions of these little messages in bottles. We are talking about extracellular vesicles, or EVs.
And, for decades, the field of cell biology largely just ignored these structures. The prevailing assumption was that they were just cellular debris.
Like a biological trash disposal system.
Exactly. When researchers saw these vesicles in extracellular fluid, the standard interpretation was that cells were simply sloughing off waste. But the sources we are unpacking today paint a radically different picture. This is actually a highly sophisticated, vital intercellular communication network.
And the mission for this deep dive into the source material is to decode the biological differences between the three main categories of this network. So we're looking at exosomes, microvesicles, and apoptotic bodies. We're going to explore how they're made, what they carry, and why they matter to you.
It's a huge topic, and the scale we are dealing with here is massive. We're ranging from structures as incredibly tiny as 30 nanometers all the way up to 5,000 nanometers.
Which is a huge variance.
It is, and that enormous size variance is really our first major clue that we aren't just looking at a single biological process. The physical dimensions of an extracellular vesicle directly correlate to its biogenesis, meaning its anatomical origin point within the cell and its ultimate physiological function.
So to map this out, we really have to start deep within the interior architecture of the cell. Okay, let's unpack this. We have to start with the smallest and, honestly, most metabolically demanding of these packages, right? The exosomes.
Right. Measuring anywhere from just 30 to 150 nanometers in diameter. And their origin story is completely unique compared to other vesicles. They don't bud outward from the cell's surface.
Right, they go inward.
Exactly. They are manufactured via an inward pathway, specifically within the endosomal network. The mechanics of that inward pathway are just a marvel of cellular engineering. It initiates when a localized region of the outer plasma membrane invaginates.
So it bends inward.
Right, it bends inward, capturing a tiny volume of extracellular fluid and cell surface proteins, and this forms an early endosome within the cytoplasm. Then, as this early endosome matures into a late endosome, the membrane undergoes a second invagination event.
Wait, so the membrane of the endosome itself starts budding inward again?
Yes, into its own hollow interior space.
Okay, so we are essentially looking at a Russian nesting doll of cellular packaging. I mean, the cell membrane is the outermost doll. The structure forming inside, which we call a multivesicular body, or MVB, is the middle doll. And those tiny bubbles budding into the hollow center of the MVB are the intraluminal vesicles, the innermost dolls.
That structural analogy captures the geometry perfectly. It really is a bubble within a bubble. And generating that negative membrane curvature, basically forcing a bubble to bud inward against the natural physical constraints of the lipid bilayer, requires some very specialized molecular machinery.
Right.
The sources highlight the ESCRT complex. That's the endosomal sorting complex required for transport. Specific accessory proteins within this complex, like ALEX and TSG101, they physically deform the membrane. And they do this while simultaneously sorting highly specific cytosolic cargo into the forming vesicle before finally pinching it off.
But once those intraluminal vesicles are formed, they're still stuck, right? Like sequestered deep inside the cell, trapped within the boundary of that multivesicular body.
Which presents a really critical routing decision for the cell. The MVB can be directed along microtubule tract straight to the lysosome.
Where it just gets destroyed.
Exactly. Its contents undergo enzymatic degradation and recycling, or, alternatively, the cell can route the MVB to the outer perimeter. And if it takes this path, the outer membrane of the MVB fuses with the cell's plasma membrane, effectively spilling all those tiny internal intraluminal vesicles out into the extracellular space.
And it is only upon that release into the extracellular matrix that these structures officially earn the biological classification of exosomes.
Right, and that entire transit process is highly regulated by a family of proteins called RAB-GT passes. The sources specifically point to RAB27A and RAB27B.
Kind of like cellular dispatchers.
Or molecular tugboats.
Yeah.
They recruit effector proteins that physically pull the MVB toward the cell periphery, and they facilitate the tethering and docking required to, overcome the repulsive forces of the two lipid bilayers, allowing that fusion to actually occur.
Because of that intense, energy-draining ESCRT-mediated sorting process, the cargo inside an exosome is highly deliberate. The cell is curating a very specific molecular payload. We see this with specific surface markers that consistently define exosomes, like the tetraspanins, CD9, CD63, and CD81.
Yes, but beyond those standard markers, our sources include a melanoma lipidomic study that really illustrates the precision of this sorting.
Oh, this study was fascinating.
It really was. They investigated two distinct human melanoma cell lines. One is LCP, which is poorly metastatic and rarely spreads to bone. The other is SKML28, which is a highly aggressive, highly metastatic line.
Right.
The researchers utilized advanced lipidomics to map the exact phospholipid composition of both the parent cells and the exosomes they secreted. They were looking for functional biomarkers of metastasis.
And the parent cell profiles were totally distinct from one another. The less aggressive LCP cells maintained a lipid profile heavily skewed towards saturated and shorter fatty acid tails, which, inherently affects membrane fluidity.
Right, but here is the twist. When the researchers analyzed the exosomes secreted by these two wildly different cancer lines, the exosomal lipid profiles looked surprisingly identical.
Which initially seems totally counterintuitive.
Right. However, the lipidomic mapping revealed a specific fingerprint. They identified a highly peculiar phospholipid called bismonooacylglycerophosphate.
Or BMP.
Or BMP, exactly. This BMP lipid was heavily enriched in the exosomal membranes compared to the parent cells of both melanoma lines.
And the presence of BMP isn't just a fun piece of chemical trivia. It functionally proves that exosomes are not formed by the cell, just like randomly pinching off a convenient piece of nearby membrane.
Not at all.
The cell is actively synthesizing and concentrating rare lipids like BMP into the exosome boundary during that complex internal assembly phase in the MBB.
It speaks to the evolutionary importance of exosomes. The cell expends a massive amount of ATP to curate these specific lipid and protein payloads for targeted, steady-state communication across tissues.
Which actually brings me to a structural question about our second category. If the endosomal pathway is so metabolically expensive, requiring multiple invaginations, the ESCRTA complex, RAB, GTPase ratting, why does the cell dedicate so much energy to this internal nesting doll process?
That's a great question.
Especially when we consider microvesicles, which are formed right at the plasma membrane. Wait, if the cell can just quickly pinch off a microvesicle right at the surface loading dock, why bother going through the whole complicated, energy-draining internal process to make exosomes?
What's fascinating here is that we are observing two parallel communication networks that are optimized for entirely different physiological states. Exosomes provide a constant, highly specific, low-volume stream of regulatory data. Microvesicles, on the other hand, function as a rapid response broadcast system.
So it's situational.
Exactly. They are heavily upregulated in response to acute cellular activation, mechanical stress, or environmental shifts like hypoxia. The cell utilizes microvesicles when it needs to rapidly offload a massive volume of bulk cargo to alter its immediate microenvironment.
And their physical profile definitely reflects that bulk loading strategy. Microvesicles, which are often referred to in the literature as ectosomes or shedding vesicles, are an order of magnitude larger than exosomes.
Right, ranging from 100 to 1,000 nanometers in diameter.
And their biogenesis relies entirely on an outward budding pathway directly from the plasma membrane.
Mechanically, though, pushing a 1,000 nanometer vesicle outward requires a radical, temporary destabilization of the cell surface. The plasma membrane is normally anchored really tightly to a scaffolding of actin filaments known as the cortical cytoskeleton.
So it's pretty rigid.
Usually, yes. To initiate shedding, the cell typically experiences a localized influx of intracellular calcium, and this calcium spike activates a whole cascade of structural changes.
Yeah, these sources detail the involvement of molecular motors like myosins, interacting with cytoskeletal components like actin and microtubules to physically exert outward pressure on the membrane.
Right.
And at the same time, Rho GTPases are recruited to the site to locally dismantle and reorganize that actin scaffolding, which is what allows the membrane to actually balloon outward.
And that physical deformation is accompanied by a severe disruption of lipid asymmetry.
Oh, this part is really cool.
It is. So a healthy plasma membrane maintains a strict separation of lipid types between its inner and outer leaflets. Phosphatidylserine, for instance, is actively sequestered on the inner leaflet facing the cytoplasm.
Kept safely on the inside.
Exactly. But during microvesicle formation, the calcium influx activates transmembrane enzymes known as scramblases and flipases.
And they collapse that membrane asymmetry. They physically scramble the lipid bilayer, pulling phosphatidylserine from the inner leaflet and exposing it on the outer leaflet of the newly forming vesicle.
Because of this direct surface budding mechanism, A microvesicle essentially acts as a localized snapshot of the parent cell's plasma membrane at that exact moment of stress.
Makes sense.
And due to their vastly larger internal volume, their absolute cargo capacity just dwarfs that of an exosome. They encapsulate a distinct, heavily concentrated profile of messenger RNA, cytosolic proteins, and signaling molecules grabbed directly from the cell periphery.
So we have the highly curated, steady-state regulatory whisper of the exosome, and then the acute, high-volume biological shout of the microvesicle.
Yeah.
Both represent a living cell actively modulating its environment.
Right.
But the EV spectrum also encompasses terminal events.
Yes, apoptosis introduces a fundamentally different set of biological mechanics compared to everything we've discussed so far.
Here's where it gets really interesting,
Yeah.
because we have to contextualize programmed cell death against standard necrosis.
Right, totally different things.
In a necrotic event like maybe driven by severe acute trauma or ischemia, the cell loses osmoregulation, it swells, and it catastrophically ruptures, spilling all its intracellular contents into the surrounding tissue. It's a very messy event that triggers an aggressive inflammatory cascade. Apoptosis, however, operates like a perfectly orchestrated building demolition.
That's a great way to put it. The organism constantly needs to turn over senescent, damaged, or redundant cells without inciting localized inflammation or autoimmunity. Apoptosis is a genetically hardwired disassembly sequence designed to safely package cellular components for swift removal.
And the ultimate products of this disassembly are apoptotic bodies, or APOBDs. These are the massive cargo ships of the EV world.
Massive, measuring between 1,000 and 5,000 nanometers in diameter. The sources outline a meticulously timed three-step mechanical process that governs their formation.
Step 1 being shrinkage and blebbing.
Right. It initiates with a cascade of executioner enzymes called caspases, which cleave and activate a kinase known as ROCK1. The activation of ROCK1 drives intense actomyosin contractility.
So the cell is basically physically squeezing itself.
Exactly. That intense contractility, combined with rapid alterations in transmembrane hydrostatic pressure, forces the shrinking cell to project spherical membrane blebs across its surface.
And following the initial blebbing, the cell transitions to the second phase, which is the formation of protrusions. The dying cell projects these elongated, microtubule-driven spikes, or beaded structures that extend violently outward from the main cell body.
And it rapidly segregates internal cellular contents into these long appendages.
It is literally dismantling its own architecture and pushing the components into these fragile outcroppings.
Which sets the stage for the third phase, fragmentation. And we actually see the return of the ESCRT complex here.
Wait, ESCRT again?
Yes, specifically a subcomponent known as ESCRT3, featuring the protein CHMP4B. But instead of forming tiny internal vesicles like it did with exosomes, ESCRT3 localizes to the narrow necks of these long protrusions and acts as molecular scissors. It severs the membrane to release these massive apoptotic bodies into the extracellular space.
Wow. And because this process is bagging up an entire demolished cell, the cargo encapsulated within an ApoBD is staggering. We are not just talking about sorted RNA or surface proteins here. Apoptotic bodies encapsulate entire intact organelles like mitochondria.
Right.
They carry massive fragments of the condensed nucleus, along with histones, the structural proteins that chromatin wraps around.
But the encapsulation of that cargo is only half the process. The overriding imperative of apoptosis is the swift, silent clearance of these bodies before their membranes degrade and they undergo secondary necrosis.
And one them bursting open.
Exactly. To facilitate this, the ApoBD actively modulates its surface chemistry to recruit professional phagocytes, primarily tissue macrophages.
The cleanup crew.
Yes, the cleanup crew. The sources describe a sequential chemical broadcast, starting with find me signals. The apoptotic body releases soluble chemoattractants into the extracellular space to establish a chemical gradient. We're looking at the release of intracellular ATP and lipid mediators like sphingosine-1-phosphate, or S1P.
And so the macrophage senses that gradient and migrates directly toward the highest concentration of ATP and S1P, arriving right at the site of the disassembled cell. However, migration is not ingestion. The macrophage requires distinct, membrane-bound eat me signals to actually initiate phagocytosis.
Which brings us back to the scrambloses and the externalization of phosphatidylserine that we saw earlier during microvesicle formation.
Oh, right. The flipping of the lipids.
Exactly. The scrambling mechanism is heavily amplified during apoptosis. The outer leaflet of the apoptotic body becomes densely coated with exposed phosphatidylserine. Additionally, proteins normally resident in the endoplasmic reticulum, like calreticulin, are trafficked to the vesicle surface.
And when the macrophage's specialized surface receptors bind to that specific matrix of phosphatidylserine and calreticulin, it triggers cytoskeletal rearrangement in the macrophage. This allows it to engulf and digest the massive vesicle without secreting pro-inflammatory cytokines.
The precision of this system is just remarkable.
It really is. But pulling back from the cellular mechanics for a moment, we need to address the practical application of this research. So what does this all mean? Beyond the fascination of inward pathways and molecular scissors, why is mapping the exact parameters of these three vesicle populations driving so much current biomedical research?
Well, if we connect this to the bigger picture, mastering EV biology is fundamentally rewriting the future of clinical diagnostics and targeted therapeutics. Let's look at the exosome profile.
Okay.
Because they are assembled via that highly regulated endosomal pathway, their molecular payload offers a pristine, high-fidelity readout of the internal state of the parent cell. This is the physiological basis for advanced liquid biopsies.
Oh, wow. So if an early-stage malignancy is developing deep within an organ, those cells are constantly shedding modified exosomes into the systemic circulation.
Exactly. By isolating those specific 30 to 150 nanometer vesicles from just a simple peripheral blood draw and sequencing their curated RNA and protein cargo, clinicians can theoretically detect tumor signatures, assess metastatic potential, or even monitor drug resistance in real time.
So it allows us to read the tumor's internal communication network long before a physical mass is large enough to be detected by traditional radiological imaging.
It's a game changer, and our understanding of the terminal end of the spectrum is evolving just as rapidly. The historical consensus, as we noted, was that apoptotic bodies were merely inert bags of biological waste waiting for macrophage clearance.
Right, just garbage bags.
But that paradigm has completely shifted. The sources demonstrate that APOBDs mediate complex downstream biological signaling. Before they are cleared, these massive vesicles actually transfer functional biomolecules, including autoantigens, microRNA, and intact genomic DNA fragments to neighboring healthy cells.
So the dying cell is actively executing a final program to influence the surviving tissue architecture.
Precisely. By transferring these specific regulatory molecules to adjacent epithelial cells and local macrophages, ApoBDs suppress localized inflammation. They stimulate compensatory cellular proliferation to replace the lost cell, and they present autoantigens in a non-inflammatory context to reinforce peripheral immune tolerance and prevent autoimmune reactivity.
It's a highly active process of tissue regeneration.
It really is.
So to synthesize the scope of what we've covered today, we have exosomes, the 30 to 150 nanometer vesicles requiring complex inward endosomal biogenesis to deliver highly curated targeted signaling.
Right.
We have microvesicles, the 100 to 1,000 nanometer structures shedding directly from the plasma membrane to broadcast immediate bulk cargo responses to environmental stress. And then we have apoptotic bodies, the massive 1,000 to 5,000 nanometer terminal packages executing a highly regulated demolition to safely recycle organelles and coordinate tissue repair.
It is a profoundly elegant three-tier hierarchy. Yet, as comprehensive as this framework appears, the sources actually suggest we are only mapping the most prominent features of a much larger topography. Contemporary research is currently isolating extracellular vesicles that entirely defy these three classifications.
Wait, really? We're finding even more distinct communication methods.
We are. Consider the recent identification of microsomes. As a cell physically migrates across an extracellular matrix, it leaves behind long tethering tracts known as retraction fibers.
Okay.
And at the distal tips of these fibers, the cell deposits large pomegranate-like structures heavily packed with smaller internal vesicles. It effectively leaves a complex spatial trail of biochemical footprints for other cells to encounter.
That's incredible.
Or look at the discovery of large oncosomes. These are massive, atypical vesicles, frequently exceeding 10,000 nanometers, that are specifically synthesized and released by highly aggressive amoeboid cancer cells to condition the pre-metastatic niche.
Pomegranate-like footprints and massive oncosomes.
Yeah.
It really highlights just how deep this field goes.
It does. It forces us to ask a larger question about our own biology. I mean, if our cells have evolved this many distinct, highly regulated microscopic languages to coordinate their behavior, from the steady metabolic whisper of an exosome to the terminal payload of an apoptotic body to the strange trailing architecture of a microsome, what other intricate biological conversations are constantly occurring in our tissues right now that our current technology simply cannot yet hear?
Wow. It completely reframes how we view human physiology. The body isn't a static architecture. It is a continuously flowing, deeply connected network of information exchange. Thank you for joining us on this deep dive into the source material. We will see you next time.